Hi, everyone, and welcome to today's Hot Topic program, Early Human Coexistence 1.5 million years ago with Kay Behrensmeyer. My name is Briana Pobiner, and I'm a paleoanthropologist and an educator at the Smithsonian's National Museum of Natural History. I'm a brown and gray-haired woman in a red shirt wearing headphones in front of a Zoom screen with an African savanna photo with grass and an acacia tree behind me. Whether this is your first time joining us for a hot topic or you've attended before, we're so glad to have you here. Before we get started, I have a few housekeeping notes. This discussion offers closed captioning. You can turn the captions on or off via the CC button, which should be located at the bottom of your Zoom interface. We're in a webinar format. We can't see or hear you. So as you have questions, please go ahead and submit them to the Q&A box, which is at the top or bottom of your screen. It looks like two speech bubbles, so that we can sort through as many questions as possible. The Q&A time really flies by. The Q&A box is also where we will share, let me try that again. The Q&A box is also where we will share any relevant links during the program, so keep an eye out there. We'll start with a presentation by our speaker, Kay Behrensmeyer, and then I'll join her here to take your questions. During the presentation, I will also write answers to some of your questions, at least any that I can answer, as well another member of our behind the scenes team, Dr. Ryan McRae. So now I would like to go ahead and introduce our speaker. Dr. Kay Behrensmeyer is Curator of Vertebrate Paleontology and Senior Scientist in the Department of Paleobiology here in the National Museum of Natural History. She's a paleontologist and geologist whose research is broadly aimed at understanding the evolution of terrestrial ecosystems over the past 300 plus million years. And she has undertaken collaborative field projects in North America, Africa, and Pakistan, ranging up and down the geological time column from the Triassic to the Pleistocene. She uses sedimentary geology and isotope geochemistry to reconstruct the physical environments of fossil-bearing deposits. Kay is also recognized as a pioneer in the field of taphonomy, which is the study of processes that affect organic remains and lead either to recycling or fossilization, and the study of land ecosystems through geological time, with a particular focus on the paleoecology of human evolution in Africa. Kay was elected to the National Academy of Arts and Sciences in 2011. She received the R.C. Moore Medal for Excellence in Paleontology from the Society for Sedimentary Geology in 2016, the Paleontological Society Medal in 2018, the Society of Vertebrate Paleontology Romer Simpson Medal in 2018, and in 2020, she was elected to the National Academy of Sciences. And now I'm really excited to hear Kay's presentation, and I invite her to share her screen.
Thank you, Briana, very much for that introduction. And I'm very happy to be here speaking with all of you about one of the projects that I've been involved with for quite some time, and that's the footprints in Kenya, which I will tell you about the initial discovery and then what's happened since that initial discovery way back in my earlier career. I'm going to try to lead everybody through some of the more complicated ways of looking at this record and pose some questions that you may be very intrigued by. So here on the title slide, we have footprints in the middle. These were some of the ones that we found initially. And then the question of who might have made them is one that we've been following for years. And we have two candidates that have fossils at the same time, 1.5 million years ago. That is Homo erectus on your left and Paranthropus boisei on the right. And these have been reconstructed by our wonderful artist here at the NMNH. I'm just trying to get, there we go. It's working now. And you can see what they might have looked like in life. But we don't know much about their feet because fossils of their foot bones have been very, very rare. So that's part of our problem. How much can we tell from looking at the footprints about whether they were different walkers, how they walked, and all of that sort of thing? Now, not all fossils are bones and shells that you might be familiar with. There are also fossil traces, as they're called in geology. Organisms leave signs in the rocks. They've done a lot of this even way back at the beginning of multicellular life, and that's a big area of study. They make burrows into sedimentary layers, and that's what you're looking at some diagrams of. And there also are tracks that are known from bedding surfaces. This is a rock from the Grand Canyon with a modern lizard on it, but it's kind of following the trail of a very, very distant ancestor that you can see that was walking on an ancient dune and left these tracks. And not only that, but we have trace fossils that are on and in bones and other organic materials. And the circle or the oval there shows something that termites do to a rib. You can imagine the whole rib. And this is something that Briana and I have a good time discussing and talking about and recognizing in the fossil record as well. Now, footprints and trackways can be preserved as trace fossils, and here are just a variety of examples of some modern ones. You can see that they are variable. There's ripple marks in the center image and some algae, and the algae actually helps to mold the tracks and keep the mud from being too squishy. On the right, you can see a trackway made on mud that was drying out. And that actually becomes important because the mud cracks form very quickly and they can disrupt and leave their own traces in ancient rocks. Now, the big question, our theme is how can you tell that different early ancestors, hominins, I'm going to be calling them, were living at the same time? Well, what do we mean by the same time? You can see on this diagram, which goes vertically from the bottom to the top through millions of years, and these bars indicate the spans of time when these different kinds of hominins existed. And you don't have to worry about all the names. What we're focusing on here are our two target kinds of early hominins, Homo erectus and Paranthropus boisei, and a time period that's right between 1.5 or 1 million and 2 million and 1.5 million. And we know quite a lot about them, but the reconstructions that you see look like whole skeletons. We actually don't have whole skeletons of both of them. We have partial skeletons, and sometimes it's very hard to relate the teeth and the jaws and all to the postcranials, which are everything below the head. So that then leads us to wonder what we can get out of the footprints. Now, the place where I've worked and where this is focused is in the Horn of Africa, in the northern part of the Great Rift Valley, and zeroing in on that red box that is in the top diagram, you can see this part of Africa, and then the smaller red rectangle is in the Turkana Basin, a place called East Turkana. And this is where I've spent most of, or a lot of my career looking at both the fossils and the footprints. There are two main areas, and on the right here, we have a stratigraphic column, a diagram. You can see that it's 0 to 100 meter scale. On the left, there are keys to the kinds of sediments. Those give us clues about the environments. And then the names and their dates. MA means millions of years ago. So at the top, we have the Chari tuff. It's a volcanic ash that can be dated at about 1.4. And the Okoti member there is the one that has the footprint records. And that in itself is another kind of problem. Like, why don't we have them all the way through? We only have them in this particular time interval. And this is a complicated diagram in a way, but if you think of you're looking at the sediments that have built up on the margin of this lake, where the red arrow comes out, you're looking at like a slice of the cake of all these different kinds of strata. The blue mean that it's lake deposits in a lake, but the brown and the yellow are deposits coming from deltas or rivers that are farther away from the center of this basin. And again, a reminder up in the top part that we have two areas, and this is a paleogeographic kind of a reconstruction of deltas that were building out into this ancient lake, Yang, um, about 1.5 million years ago. And this is where the trackway records are. We're fortunate that we're in a rift valley because it continues to subside. So the sediments can build up to great thicknesses. And then because there are earthquakes and there's a lot of tectonic activity, it's called in the Rift Valley, some of these are lifted up so that we can look at them today. And if you can imagine the modern lake on the left there, and here we have a delta today that's building out and would create lots of nice soft sediment places where footprints could be preserved. And we're lucky actually to have the modern analogs, we call them, of where animals leave their prints, and examples of the modern hippos that are there's still a few in Lake Turkana. And when I started working on this project in the 1970s, there were lots of examples of their tracks that we could use for comparison and thinking about what conditions would preserve really nice tracks. You also had to be careful not to encounter the hippos themselves in those days because they can be quite intimidating and dangerous. Now, the story of our initial find, I was out with colleagues in 1978. We were actually just trying to record the body fossils, as they're called, the actual hard fossils of the bones and the teeth. And we were focusing on one layer, one stratum, one thick stratum that was about 1.5 million years old in these different areas, Cubifora and Illuret. So we were digging to expose the sediments, and this is my dear colleague Muteti Nume, and you can see that he's dug this beautiful trench, and there are some interesting features in near the top of it. And we realized when he had dug through these very carefully and neatly that these were likely hippopotamus prints. And then you can see the very thin beds, and then at the top there's a gray unit, and that fortunately is a volcanic ash, so it can help us date this site. And then I thought, well, let's see what these footprints look like from the top down. So I went away and after giving instructions, and Kamolo was very carefully brushing off 1 square meter, as you see him here. And when I returned from doing more surveys, I looked down, and lo and behold, there were actual hominin footprints. I mean, they were just completely convincing, and amongst the hippo prints. So we were incredibly lucky that we happened to expand our excavation into a place where a hominin long ago had walked on the mud, and actually it looked at one point like it had fallen into one of these deep footprints of a hippo and then managed to catch itself so it didn't fall all the way down. And of course, we joked about, well, maybe if it did fall down, we'd actually get a fossil face print. But unfortunately, that has not happened yet. Anyway, Kamolo was very excited about this, and we knew that we had something that we'd have to work on in a much larger sense. And here's where the date, and later, we didn't know this at the time, but later the date was about 1.4 million years. So in 1979, we went back and expanded the excavation. You can see we're looking down there. The tripod thing looking is a camera. We didn't have drones or anything like that at the time, so we were lifting this camera up to get good views of the whole excavation. And we put black sand into the footprints so that they would show up in the photographs. And this is then from the publication in 1981, a map of these prints. And you can see they're hippo trackways. There's a big bird print right in the middle there. And it was pretty clear from the footprints of the hominins that it had come out of the water, or at least sloppier mud, into a much firmer substrate. So this was the beginning. This was exciting, but it happened in 1978, and just by chance, the same year, a place called Laetoli near Olduvai Gorge down in Tanzania, uh, hominin trackways were also found, and they were much, much older. So ours kind of got in the shadow of these, which turned out to be, well, and their later ones discovered really wonderful and exciting evidence that at this time period, the hominins were bipedal. And also, this looked like a group of three with a young one walking along. So we were not involved with that. I was in the Turkana Basin and continue to be there. But I think, both these finds began to open up people's eyes to the fact that you could get a lot of information from fossil trackways. So we went on at Turkana, various people doing discoveries and excavations of more hominin prints. Here you can see the prints of a lot of them on one of the surfaces, but it wasn't just one surface. This was up at Illirat. The previous ones I showed were from Kubifora. So, suddenly we were finding hominin prints at different levels and lots of different surfaces. These are about the same age, though, about 1.5. And here was a whole group of hominins moving across the surface. So by 2012, we had a total of 73 footprints on different surfaces, 2 of them at Illirat, and that's just continued to expand, and more publications are in the works. But let's get back to this question of who made the tracks. Because we have 2 candidates, and we could assume that maybe it was Homo erectus that was walking around there, but why not Paranthropus, which was also bipedal? So this brings us to the paper that came out in 2024, where my colleague Kevin Hatala, who is a paleoanthropologist and anatomist who knows a lot more about foot anatomy than I do. I was providing a lot of the geological context. And here are 2 photographs of feet, footprints that look, you can see they're kind of different. But is this because they were just deciding to walk differently, the same kind of individual, or that they were getting into squishier mud, or, what's going on with this? So how could you tell? So the track surface had multiple footprints, one nice long trackway that you can see with the dark blue prints that was clearly one individual walking across the surface. And below there's the 3D scan. Kevin Hatala is also an expert at doing that. So that's giving a sort of topography with the colors. But here were 3 more prints that were different. That is, they weren't in the trackway, and they looked to him at least to be different in shape. I was able to do this stratigraphy on this. That is the layers of different kinds of sediment. You can see another one of these nice trenches that the Kenyan excavators are so good at. And you can see here that actually there are 5 different levels that in cross-section have evidence of tracks. And just one of these has been excavated, and that's what this long trackway was discovered at. So the track making candidates, just to review, here's one of these tracks with our 2 possible track makers. Okay, so Kevin decided he would really need to look at what happens when feet hit mud of different kinds. He did lots of experiments, and he worked with these 2 colleagues, and we try to give a lot of credit to that. And you can see also I've put in the publication information because all of this needs to be documented, and we need to be accountable for where the science is published. So in this case, you can see a little video showing what they were recording, the way that the foot hit the substrate, different kinds of muds were used, and all of that then became analyzed as art volume, and it's called relative art volume, and that can be different shapes for different kinds of walking and morphologies of feet. And then Kevin did a lot of work on modern humans, some that had worn shoes their whole lives, some that had not. And every dot you see here in this sort of violet plot of the relative arch volume on the y-axis, the left, and the depth of the track is from a modern human sample. But of course, we're the genus Homo, and we have a very nice high arch. So how do the arch morphologies in these tracks compare? Well, this diagram is just going to show you three of those. One, the one down in the lower part, or C, is from one of the tracks in this long trackway. And that falls right outside of the range of anything that modern humans make, or other examples. And Kevin's also put in Laetoli, those more ancient footprints, which are falling off the sort of light purple. But the two of the tracks, the isolated tracks, and actually all three of them fall into the more human kind of range of arch volume. So there are arches, and you can see that in track H3. The arch volume is apparent in that greener color between the heel and the toes, whereas in this long trackway, you just see it's a pretty flat foot. So that's the evidence for two different arch morphologies. And there's even more evidence from the angle of the big toe, which you might have noticed before. This is Homo erectus. At least this is what we think, because it's more like Homo. And the toes, you can spread them when you're walking on mud, and it may vary a little, but it's nothing like this morphology, which we now think is the Paranthropus morphology. And it has a low arch to almost no arch and this angled toe, compared with the arch and the straight toes of Homo erectus. Now, let's not say that it's all for sure yet, because there are a lot of people who would like to see more evidence, and we're working on that. But this is pretty strong and convincing evidence that there are two different morphologies that go with the two different hominins. And we need foot bones, and now there's some new ones that are being described. I can't say anything about them that may help to support these hypotheses. So more questions. Did the different hominins make the tracks at the same time? So this is, maybe they interacted, maybe they didn't. How can we say anything about that? And another question is how they interacted with other animals that are on the lakeshore, like the giant storks and the hippos. So Kevin did experiments, and along with other people, another collaborator, Neil Roach, has worked with us on this, and in the modern lake, just recording what happens to a footprint, the same footprint, after 0 days, 1 day, 2 days. You can see actually an antelope came along and stepped on that one. 3 days, and by 4 days, it's basically not going to tell you much of anything about who was there. So there's a very short time period where the footprints need to be buried, or they will no longer be informative. So that means that the footprints that are found on the same surface have to be less than 2 days, and probably much less than even 1 day. So this is a reconstruction then, following upon that. Assuming that the tracks on the same surface are going to take it less than a day or less than 2 days for sure. That still doesn't mean that the animals like these reconstructed on this modern lakeshore, a zebra, an antelope, hominins, a hippo, would encounter each other because there are a lot of hours in the day. But in an ecological sense, this means that they were contemporaneous in a way that you could never show from just those fossils that I indicated in the beginning on those bars on a time bar. They were, if not interacting with each other, they were using the same environments at the same time. And here are some of the other animals that we've recorded: rhinoceros, stork, hippo, crocodile, bovids, or the two-toed ungulates, different kinds of birds, even elephant. And the proportions of these in the different places on these two paleodeltas that's subject to a lot of interest and more research. The excavated surfaces, we now have 27 of them, at least when I made this diagram. And what's very interesting is that there's so many at Illirat. You can see the hominins are pretty common, tracks per square meter average. Bovids and pigs, our suids, are the most common. They like to go down to the lake edge to drink. And birds are pretty common, and they're different in the two areas as well. Something surprising here is maybe why there aren't more crocodile footprints. We haven't found any other primates like monkey footprints either, even though they're known to go down for water. And they were there in the fossil record with the bones. So there's lots more to do of interest here. And we are actually heading back to the field in July to look at more of this stratum that had at the bottom, that 2024 paper, the two hominins represented on a single track surface. And then up at the top, we have a paper in review proposing that those footprints are only Paranthropus at the top. But of course, the time doesn't necessarily mean anything. They were there, both there for a couple of 100,000 years that we can date in this long sequence of strata. So 80,000 years in this particular one, which is in Kubifora Area 103. And look at all these. These are footprint horizons that we potentially can excavate and find more evidence of how these hominins were using this landscape, and why did they keep coming back to the to make footprints in these lake margin environments. Most of the strata do not record footprints, so it's a very selective sample, but they kept coming back to it over a couple 100,000 years. That we can document for sure. So the hominin fossil footprints in general are more common than people realize. They record the ancient species in real time that we can all relate to, like going to the beach and seeing all the footprints. And that means what's called ecological sympratry. They can say that they really did exist on the landscape at the same time in an ecological sense. We can use the skeletal fossils and we hope, more foot bones to expand the understanding of the anatomy and how they walked and their ecology. And then, the behavior. What were they doing that brought them to the lakeshore over all this time? Were they accessing some kind of resources? Were there different resources? Did some of them catch fish? Did they eat the plants that were growing there? Maybe we'll get some answers with more field work. I'd like to thank people then and now, the field crew, with myself on the left there, looking quite different from 2023. Also on the left with our wonderful colleagues and experts, and a lot of people who've supported institutions. And we couldn't do it without all of the contributions of these folks, and especially my two colleagues who are contributing the paleoanthropological side of the story, Kevin and Neil. So I think I might have come in at the right time.
That was perfect.
Uh,
Wonderful.
I will now stop sharing.
Yep,
And
I think you have already. Perfect.
oh, okay.
Great. So wonderful. Thank you. That was fantastic. And we have some questions that have already come in. So I'm just going to go ahead and jump in. And actually, the first two questions are very similar. I think asking the same thing. So I'll read them both. Toby asked, is it possible to tell if the individual was rocking or running? Um, and Walter asks, is it possible to ascertain whether the footprints show a hominin walking at a casual or slow pace, or whether they were running, such as in an animal hunt? And then I'll ask the second part of Toby's question after this.
Okay, those are great questions. And I will do my best to say what Kevin and Neil would say. That is that you can tell by the stride length that all of them so far were walking, that we don't have any evidence of running, but there are more or less deliberate walkings across these substrates. And where I showed the groups of prints, it's pretty clear that there were different people walking along together
Mhm.
at Illeret, and so they were interacting in a way that we can speculate about, but they were not walking fast. The single footprint track in that 2024 paper, that's the one that is Paranthropus, seems to be striding rather purposefully across the substrate, even though it was fairly soft and
Mm,
very directly, and it would have been going sort of parallel to the shore, I believe.
mm,
Um, but and the other interesting thing I didn't mention is that there are no mud cracks in any of these substrates. So they were walking in shallow water, not on the drier surfaces. So that's another intriguing thing. And maybe you wouldn't want to run on in shallow water. Or if you did, you might completely destroy any tracks that, by
that's
splashing.
true.
So,
And actually, that really leads in well to the second part of Toby's question, which was, would individuals run through wet areas or walk through them? I don't know if we have any evidence of that as far as what we can tell from the like fossil footprints.
well, I guess it depends on whether they were having fun or not.
That's right,
like
exactly.
like our modern people do at the beach. I mean, I
Yeah.
have I have sort of fantasized about, well, it's nice to put your feet in the water if it's a hot, open place. And
Mhm.
yet there seems to be more deliberate activity through walking. So uh,
Mm.
I I think they might have been kind of serious also about looking out for hippos and crocodiles and other threats that could have been also congregating in the same area along the lake.
Mhm. Yeah. And I guess like what you were saying before too, is if the footprints were made in really wet sediments, they might not preserve anyway. So I think we also have to think about kind of that sort of preservation bias in a sense. Um, and I see Ida wrote, or running from being eaten. And certainly, yeah, like you were just saying about hippos and crocodiles, which were are threats today and probably were threats in the past.
I will tell a little story. I actually consulted a tracker, um, someone who was knowledgeable about tracks, and when we found that original trackway in 1978, and his interpretation is that because of the angle of the tracks toward the um, the end of it that was coming out of the outcrop, which was kind of tilted, he thought that the individual had looked to the left and was being cautious about watching something.
Cool.
Yeah.
Wow.
Um, yeah, we
Um,
could we could probably get expert trackers to do a lot more with these.
that
And
would be really neat,
yeah.
actually. Okay, I'm going to go to one of our behind the scenes questions now. And so this question is, trace fossils like footprints tell us where organisms actually lived, while fossils of their body parts like bones tell us where they died.
Hmm.
For hominins or for other animals at the same time period, is there a difference between the depositional environments of trace fossils like footprints versus body fossils or bones? Or how would researchers even go about investigating that question?
Ha, wonderful question, and something that's like guided a lot of my career is figuring out the difference between where animals lived and where they died and got preserved and buried. Interestingly, in this unit, which is called the Okoti member,
Hmm.
um, there are a lot of body fossils, and
Hmm.
the layers are not exactly the same, although sometimes in some of the footprinted surfaces, there are bones that were brought in by currents that are trapped in the footprints.
Huh.
We found that at the lower part of Kubifora Tuff, it's called, the
Hmm.
same level where the 2024 publication showed those. And actually, there are hominin remains almost like immediately above the track surface uh,
Huh.
there, which are in the process of being published. And they're Paranthropus, I guess, reveal that. So it can be very closely juxtaposed in the right circumstance. And so we're going to try to make the most of it.
Uh-huh.
But, there are a lot of tracks on a surface and maybe only one or two bones of a particular species or a
Hmm.
fragment. And so, in order to understand enough about the anatomy of the animals from the bones,
Hmm.
you have to go looking in a lot broader places and
Mm.
different strata and put together the samples from different time periods and places. And so that automatically means you are incorporating a lot more time
Oh, yeah.
from so you get a big enough sample of the fossil remains. But I think we're starting to try to compare what's going on with those fossil remains, the bones and the teeth,
Hmm.
same unit,
Hmm.
and the trackways. And there may be no better place to do it than this. Laetoli actually has in Tanzania that they have a lot of fossils in the same beds as well. Uh,
Cool.
but generally, people tend to study one or the other. So
Yeah.
we have to,
Yeah.
but great question. And you can just see the potential for the future research with.
Yeah. And actually, I have a follow-up that I was thinking as you were giving your answer. Are the same sorts of like preservation environments, do the footprints get preserved in the same sorts of depositional environments that body fossils do? Or is there going to be some sort of bias? Like footprints are always going to get preserved. Do you always need water? I don't know. Like, would you expect to find differences in the first place?
Yes, because the conditions for preserving the footprints are so specific. You have to
Yeah.
have the mud. And the mud can't be too muddy or, it
Mm,
needs to be plastic. The feet come along, they make their impressions. And then within a day, I mean, which is what we showed with those experiments, you have to have it gently filled in, not a lot of energy
mm,
on the shore. So it has to be a shallow and flat kind of shoreline with gentle waves that bring in sand. And that's what happened with again and again. So
hmm,
it's a very flat kind of delta
hmm.
landscape. But the bones can be preserved in a lot of other different circumstances because they're kind of robust and they can survive being transported by a river or, more energy. So uh, the options for preserving the skeletal material are many, many more channels and floodplains and lake margins and deep lakes and no,
Yeah.
it's just a narrow band of the right conditions along the lakeshore, and not the whole lakeshore, but places like the delta where it's building up and it's also subsiding. So you
Huh.
don't, if a storm comes along, it doesn't destroy everything because it's subsided out of the way.
Okay. So yeah, very specific. We're lucky to have any footprints, I guess, in some
Well, and
ways.
that's what I said that the other parts of the Cuvifora Formation, we don't
Hmm.
seem to have those conditions. I think it
Yeah.
also has something to do with the volcanic ash that got worked into the muds and maybe it was a time when a lot of sediment was able to build up quickly.
Yeah. Okay. Here's another question from Walter. Can the footprints tell whether the two hominins kept a distance versus like traveled separately to keep away from each other? Thinking about whether there was some kind of socialization, he asks, especially for hunting.
Well, you've given me the idea that maybe in the future we will find trackways that have the different morphologies, but they're going along together.
Yeah, that
It's
would be the,
possible. That
yeah,
is possible.
yeah,
I mean, the story has been for decades that you couldn't have two different bipedal hominins living at the same time and place because they would compete with each other. And
right.
that's sort of our
Default
default
assumption.
is that they're going
Yeah.
to exclude one another. It's called competitive exclusion.
Hmm.
But right now, all the evidence points to the fact they were inhabiting the same terrain. They
Hmm.
were at the same time, and for hundreds of thousands of years, they were not competing in a way that eliminated one or the other. So they might have
Hmm.
different resources. They might have
Hmm.
interacted the way different kinds of primates on the landscape do, baboons and vervet monkeys. And, I mean, you can
Hmm.
imagine that fairly closely related animals of all kinds get along
Yeah.
fine as long as they have enough to eat and other resources. So,
Hmm.
um,
Yeah.
but there may be, that would be the next big exciting thing, I think. The next hot topic,
Exactly. Will be,
even
yeah.
hotter topic.
I
It,
think about that sometimes with, in landscapes like the one behind me where you see like two species of zebra or multiple species of bovids. I think, well, they seem to coexist just fine. So, um, especially if they have like a little bit of different niches or different food or something like that. So,
but also, lions and hyenas and the big predators do have much more issue with each other. So if there was more hunting in the mix,
mm,
going on, or use of scavenge resources, or things that were
hmm,
at a premium for one or the other or both, then,
yeah,
we don't have any evidence yet in the trackways of tool use, for instance, or, dropped. I guess that's another thing you could imagine, drop an artifact.
exactly.
But or digging with your hands, maybe to get resources like tubers of plants and water
Hmm.
plants. So
We should just look for like digging prints or something.
yeah,
And actually, this leads me to another behind the scenes question that we have is so is it possible for other locomotor behaviors, or I guess other behaviors in general, like pacing, gathering, other movements to leave other traces like footprints and sediments? Um, or is walking somehow fundamentally different and more likely to leave footprints that stay long enough to get fossilized? I if a hominin were to like fall down or fall over or like what do you want to get will you get like a mud angel? I'm joking. But if if there's something else that presses into the surface, um would we recognize that? Do we see that in other animal traces? So I'm thinking of, I don't know, like, do you see I don't know. Do you see things other than locomotion in trace fossils?
yes. And the answer is beak marks from birds.
Oh, cool.
Yeah, and it's entirely possible that one of these hominins would put a hand down just for fun or to grab something. And I think
Mm-hmm.
the excavators are very careful. And if this is not hard rock. It's
Mm,
mud, sort of
yeah.
solidified mud. And the
Hmm.
sand is easy to brush off of it. So the details are there, but you have to be really careful.
Hmm.
Maybe we wouldn't recognize something
Yeah.
like that.
Yeah.
Possibly, but if like a walking stick, for instance,
Yeah,
you know,
exactly.
could
Mm-hmm.
that's a possibility.
Mm-hmm.
Um, and I guess even if they sat down,
Right. If they
um,
sat in the shade or if they, sat next to they were making stone tools or something. I don't know. Yeah.
well, it would have to be in a pretty sloppy place. So
Oh, yeah, that's
yeah,
true.
no.
Yeah.
But I think this is a whole new arena of observations and realizations that we're opening up. So um, there'll be surprises. I suspect
Yeah.
there will be some surprises. We have one carnivore mark, I think, so far,
Oh,
one
cool.
paw,
Hmm.
maybe a jackal trackway.
Hmm.
So you'd also expect some of those, no snakes, but the
Okay.
crocodiles, um, there should be more of them
Hmm. Yeah.
based on the modern uh, environment.
Crocodile prints, footprints, you
Yeah,
mean? Yeah.
or
Hmm.
belly prints. They
Oh, yeah.
they
Yeah.
like to haul out and rest and sun themselves, and they
Yeah.
do that now. So,
Yeah.
um,
Yeah.
well, good question.
Yeah. Here's another question from Toby who says, I have a foggy memory that Lake Turkana is high in fluoride. How might that impact like food sources or anything like that from the lake? Or maybe like what might be attractive to the hominins in that kind of environment, maybe?
Fluoride, well, maybe today you see the lake in the past, one
Hmm.
and a half million was probably a freshwater lake. Today,
Okay.
the lake is more saline and alkaline, and it's a
Hmm.
closed basin. And it's evaporating. The climate was different in the past too.
Hmm.
Chlorine is generally associated with a lot of volcanics, so maybe, yeah.
Hmm. Okay.
But I suspect that the drinking, today the animals don't like to drink because it's so alkaline.
Huh. Okay.
They
Yeah.
will, but
Hmm.
and sometimes they have to, but we
Hmm.
humans, the researchers don't want to drink that water. It's just
Okay. Yeah.
um, it doesn't taste good and it's not very good for you. So um, the water itself would have controlled the kind of plants. So freshwater papyrus, for instance, is
Hmm.
other kinds of potentially edible root type plants would have been only there if it was pretty fresh water.
Okay.
But I don't think there were any trees nearby,
Yeah.
or
Okay.
because there are no evidences of trace fossils of roots are another thing that we can see, and
Okay.
we don't have evidence of roots or grass.
Okay.
That shoreline I showed with the hippo tracks, the grasses grow over those tracks very quickly,
Hmm.
and so you need fresh sediment just deposited to get the kind of tracks that we see. And if they're there for very long, even if they're um, in good hard mud, the grass is going to grow over them, or
Yeah.
the next prints are going to be under grass. So
Okay.
I,
Um, so speaking of this sort of leads into a question by Patrick, who just wrote footprints found today in stone layers with a question mark. And I think maybe he's asking about sometimes you see, footprints of I don't know, ancient humans or other animals that look like they're very much in like hard stone layers. Like, how does that happen? Because it sounds like the footprints you're describing, these footprints are in like softer sediment. So how does that work with those ones found in very hardened layers?
it all depends on the kind of sediment.
Hmm.
And Laetoli, it was the kind of sediment from a volcano that set up almost like cement very soon after the tracks were made. Initially,
Hmm.
and Brianna, you've been working on, similar kinds of things, more young deposits, but where
Hmm.
you get this volcanic ash with a lot of carbonate in it, and
Hmm.
you have rain on that, um, it it just solidifies
Hmm.
very.
Okay.
And in other cases, if you have the sediment buried and the groundwater is going through with minerals underground, and that
Hmm.
solidifies the rock so that
Hmm.
when it erodes out, the tracks are obvious, and those
Okay.
are well preserved. And very ancient ones, as well as fairly recent ones, that can happen. Maybe if some of our footprint horizons are buried and dug up by paleontologists in a couple million years, they will be hard rock too. But
Yeah.
at this point, the only hard rocks in these strata are where there are sandstones that have um, been penetrated by water with a lot of minerals. And so the
Okay.
sandstones get hard because the minerals are precipitated between the grains, but in these muddy sediments, the water doesn't get um, to go through with the minerals so easily. So
So it sort of stays there and keeps it
yeah,
soft a little bit. Okay.
but fortunately, the sandstones kind of make sandwich bread parts, and the fillings are softer, but they're preserved and they're hard enough that
Yeah.
they don't fall apart right away. But we have to do all of our recording with the 3D imaging now.
Oh, okay. I was going to ask actually sort of what is the process at this point of sort of uncovering and excavating? Like what does that look like when you're actually doing this investigation?
Well, fortunately, we've reburied a lot of these surfaces.
Okay.
Can't we did a model of the original one with latex and then plaster backing on it, and because in '78 we didn't have many capabilities that we do today for the
Mm-hmm.
3D imaging. I did take stereo photographs, but um, we couldn't obviously take it out because
Yeah.
we tried putting hardener on it, and that was not a good idea because then the moisture underneath comes up and the
Oh.
surface is solidified, so it cracks. Yeah.
Mm-hmm.
So it's mainly done through the 3D imaging, and then
Okay.
models. You can make 3D prints, and we've done
Mm-hmm.
that of that. But then to preserve the surfaces, we just bury them again.
Okay.
And Kevin, in 2023, we excavated the same surface that had been buried in 2016, and before that had been buried in 1979, and it was in really great shape.
Oh, nice.
You know,
Okay.
so the burial has to be careful so you don't include a lot of seeds in the dirt that you're putting on top, because that happened at Laetoli, and the roots will go for anything.
Oh, yeah.
Yeah. So, but yeah, it's a special kind of curation problem for
Mhm.
not being able to collect things like you can collect bones and take them back.
Yeah, protect them. Exactly. I'm going to go to another behind the scenes question, which is so we can even see in many ways those species level or genus level morphological differences. Do you know, and maybe this is more of a question for Kevin and Neil and the foot people, but can you pick up finer grain differences like ages and sexes and,
Mhm.
sort of senescence, like, maybe differences in gait as you get older? And what features distinguish those sorts of things from the species level differences?
Well, that is a great question for Kevin or Neil or the anthropologists. But
Hmm.
one of the interesting things about the tracks we've uncovered so far, a lot of them, is that they're very few small ones. Like
Mm.
at
Ah,
Lycoli,
okay.
there's clearly a child walking along,
Hmm.
and I don't recall that there are any small individuals. So
Hmm.
like that, there may be smaller ones that could be female, but a lot of them, I think the anthropologists have said they're probably male and male group,
Okay.
and um, you could potentially, with enough of these trackways, get some idea of the individual's age potentially. I would
Hmm.
think we could
Yeah.
certainly do that today. I mean, our walking, it's well known that the way we walk is one of our distinguishing characteristics. So, um, but we'll need a bigger sample to do that. And yeah, the tracker folks, I'm sure, if we got some of them gathered around, they might have some interpretations of that. Was it tired? Was it,
Yes,
on its way somewhere with purpose, or was it
exactly.
meandering around, not knowing what to do?
I'm always thinking at some point we'll find like just a small set of kids' footprints. Like you put a kid down, you pick a kid back up and carry them. So I think that
Yeah,
would be great. So
or a heavier, a small footprint, but clearly heavier because they're carrying something.
yeah, yeah, exactly. All right, we have time for one more question, and I'm going to go to another behind the scenes question, which is that many people may not think of taphonomy as applying to trace fossils at first, but um, so this goes to where and how did your scientific career in taphonomy begin? And what is it about the taphonomy of trace fossils or body fossils that sort of draws you in more, or is it just any kind of taphonomic question?
Oh my, that's going to be hard to answer in a short period of time. I guess a lot of my interest is that in taphonomy is the the transition of living remains to the dead and then into the fossil record. It takes expertise in geology and biology and ecology, and
Hmm.
it brought together a lot of the disciplines that I was interested in because I didn't want to just end up. I mean, I love geology and that's where I started, but um, it is a fascinating kind of quest to look at things from different viewpoints and
Hmm.
understand what happened to any fossil has a story, any footprint has a story too about the preservation, and to understand that and be able to reconstruct it. It's bringing the past to life in a lot of
Hmm.
different ways. It's uh, in this case, of course, who done it for the who did the footprints is also brings in the anatomists and that kind of experimentation as well. But the disintegration of the tracks,
Hmm.
that is track taphonomy and the rates at which it happens. So, I look at how fast bones disintegrate on the landscape and what proportion of them get buried, and then how do they survive to actually become mineralized and fossilized, and how does that tiny little sample of the past faithfully or not so faithfully represent what actually occurred. I guess the curiosity is about um, what happened in those times and
Sort of the
how much can we
processes.
say? How much
Yeah,
can we say about it? Um,
yeah.
yeah, it's endlessly fascinating.
Yeah. Excellent. I think endlessly fascinating is a perfect way to wrap up, actually. So I will conclude today's virtual program. So please join me in the audience in thanking Kay for sharing her work with us. I'd also like to give special thanks to those who made this program possible, to our behind the scenes team who helped sort through your questions, including Dr. Ryan McRae, to our donors, volunteers, and viewers like you. And finally, to all our partners who help us reach, educate, and empower millions of people around the world today and every day. We thank you. Next month, we'll be featuring University of Reading lecturer in archaeology, Anamieke Milks, on April 16th. So you can grab a ticket in the link in the Q&A and find out more information about our website and in our emails, in your email, sorry, about that program. We have put a link in the Q&A where you can find information about our other upcoming programs and how to sign up for the museum's weekly e-newsletter. Um, that's the best way to stay informed on upcoming programs and to learn more about the museum's research and exhibitions. Right after this webinar ends, you'll see a survey pop up asking for some feedback about the program. Please take a moment to respond. We're very curious to know what topics you might be interested in seeing for future programs, and we appreciate your input. So again, thank you to Kay, thank you to the audience, and we will see you next month.